We go further on with the next talk, Dr. Daniel Carter from Barcelona, and he is here the junior leader in research fellow molecular bionics laboratory institute of bioengineering of Catalonia.
Thank you very much. Yours. Good morning. Thank you very much for the opportunity to share the work that we're doing in sunny Barcelona and the Institute of Bioengineering of Catalonia, where we're working to generate artificial brain targets. And so basically, why do we need artificial brain targets at all? And the reason is because of the presence of the blood-brain barrier, which, as we all know, is made by the highly specialized and highly impermeable endothelial cells lining the brain vasculature. And this high impermeability is in large part due to a very low rate of endocytosis. And I will touch upon this later on, but for now, I'll just say that this high impermeability, of course, protects us from toxins and other pathogens, but also prevents the delivery of medicines to the brain. So, as we all know, what nanomedicine tries to do is encapsulate the therapy within a nanoparticle and decorate the nanoparticle with a ligand that binds to a natural target, such as transferrin receptor, LRP1, or the glucose transporter. However, a big problem is that these natural proteins are also expressed in peripheral organs and in circulating cells. Therefore, you end up with increased off-target delivery. And these are just 3 examples of very good papers where they successfully increase delivery to the brain using nanoparticles functionalized with antibodies against transferrin receptor, a peptide mimicking transferrin, or LRP1 peptide, so AngioPep. And you can see that they increase brain delivery, but also they increase delivery to, in that case, spleen, the lung, or the liver. So how can we overcome this off-target delivery? And our idea is to, instead of using natural targets, shift to generating artificial targets by exploiting the impermeability of the blood-brain barrier itself. So, as I mentioned to you, the impermeability is in large part due to a very low rate of endocytosis, and I won't go into the details, but basically you can see that in animals where the physiology of the BBB is impaired, there's a higher rate of endocytic vesicle formation, and also in vitro, if you knock out the phenotype of brain endothelial cells, the rate of endocytic vesicle formation is much similar to the peripheral pancreatic endothelial cells, but if you rescue the BBB phenotype, this endocytic rate is lowered. So how can we use this to our advantage? And the main idea is that we would attach a molecular probe, a molecular tag, to the surface of endothelial cells throughout the body, which, because of the higher rate of endocytosis in the periphery, it is endocytosed and therefore removed more quickly, so that after a certain period of time, it is only displayed on the vasculature of the brain. And then by generating nanoparticles, which are functionalized with a linker, which can efficiently recognize these artificial targets, we should be able to have no interaction with peripheral vasculature and selective targeting or selective increased delivery to the brain. So first, we wanted to corroborate that this actually could work, that we could exploit the endocytic rate, the lower endocytic rate to generate artificial targets. So basically, we attached a biotin molecular tag to the surface of brain, liver, or lung endothelial cells, and we incubated them at 37 degrees to allow for the endocytic removal, and then quantify the retention of the biotin tag by attaching evident FITC. And you can see that in the lung and the liver, here in the green and the red case, the endocytosis is quite fast compared to the brain endothelial cells, where the retention of biotin targets is more slow. And this differential endocytosis can be exploited to increase the targeting to the brain over these peripheral organs. This is in vitro, and therefore we wanted to see whether this strategy could be used in vivo. Obviously, we could not use biotin NHS like we did in vitro, so we opted for using a biotin-related anti-PCAM1 antibody as the molecular tag, and avidin functionalized polymeric polyionic complex micelles as the nanoparticle. And you can see that, okay, I will skip this for time's sake. So we carry out this experiment. Basically, we injected the biotinylated antibody into mice, and then we waited a certain time interval before we injected the evident functionalized nanoparticles. And you can see that at the short time interval of 15 minutes, most targeting is into the lung, but we also have targets into the brain, the heart, and the pancreas, meaning that the antibody is being retained on the vasculature of these organs. However, as the time interval increases, the targeting to the peripheral organs steadily decreases. But what you can see is that the targeting to the brain remains fairly constant, so that after an 8-hour time interval, the only significant targeting that we see is to the brain, indicating that the antibody has been selectively retained on the brain vasculature, thereby acting as a brain-specific artificial target. This shows that we can harness the low endocytosis of brain endothelial cells to generate artificial targets to increase nanoparticle delivery to the brain. However, then we wanted to know whether this strategy could be used with any type of cell membrane protein, since the endocytic rate might depend on the protein function. So we repeated the experiment, but instead of using PCAM1 antibodies, we injected transferrin receptor antibodies. And you can see that in this case, the targeting to the brain decreases at a faster rate than targeting to the lung, so that at the 8-hour time period, we lose the specificity of brain targeting, meaning that, no, we cannot use any cell membrane protein to apply this strategy. So we need to probe the endocytic rate of individual cell membrane components to be able to identify the molecular probes or the molecular tags that will allow us to apply the strategy. And to that end, we screened libraries of phage display peptides, and we did this even though it's an indirect method of quantifying cell membrane component internalization, and we did it for two main reasons. One, the high variability of the peptides in the library allows us to bind to virtually all cell membrane components, not just the proteins. And two, by identifying the peptide, it gives us directly a molecular probe that we can use to apply this strategy. And so, in a nutshell, what we did, we screened for two types of peptides, either the peptides that bind to lung, liver, or brain endothelial cells. This is in vitro. Or, and we call this the binding population, or the peptides that not only bind, but after a certain period of time are selectively retained on the surface of each endothelial type, and we call this the retained population. And so you can see three interesting points from the populations that we identified. One, the binding population is highly heterogeneous, which reflects the large number of binding sites that the peptides can have on the endothelial cells. But the most enriched peptides are shared by at least two of the endothelial phenotypes, which shows that the cell membrane composition, even though it's heterogeneous, it is strongly conserved between endothelial cells. And thirdly, despite this similar composition, the retained peptides are highly specific for each endothelial phenotype, meaning that the endocytic rates of individual components are highly dissimilar in each phenotype. And this we can take to our advantage. And the most promising peptide that we found was this green peptide, CFAC. These are 12 amino acid peptides, but I'm just showing the first 4 amino acids for convenience sake. And so the most promising was CFAC. As you can see, it binds to the liver and the lung, but it is removed from the retained population of the liver. It's reduced in the lung, but it's highly enriched in the brain. Therefore, we wanted to see whether this peptide allows us to apply this strategy. And so we synthesized the peptide. We attached the biotin molecular tag to it. And we treated brain endothelial cells or liver endothelial cells, similarly like we did before. And you can see that in this case, we were using the BAN3 cell line. The endocytosis of the CFAC peptide is fairly constant in the liver endothelial cells, while for the brain endothelial cells, there is a fast decrease in retention, so fast endocytosis in the first hour, but then it stabilizes and remains fairly constant for about the next 8 hours, meaning that as, again, the time interval increases, the brain-to-liver ratio increases, meaning that we are able to increase the targets into brain endothelial cells. And we also repeated this with primary endothelial cells, and we see a similar pattern. Then, an obvious question is, okay, your peptide is being retained on the cell surface. Can it actually be used to deliver cargo intracellularly? So we did this experiment where we attached the peptide to brain endothelial cells, with the biotinylated peptide, and then as a model protein, we attached avidin, FITC avidin, and we monitored the localization of the avidin protein with time. You can see at the early time points, the avidin signal in green is fairly evenly distributed throughout the cell membrane, but as the time increases, this cell membrane localization starts to be disrupted, and you can start to see how it's beginning to be endocytosed into vesicular compartments. So by the 4-hour time point, we see no cell membrane localization, and it's mostly intracellular. And we also saw this, I should have said that this is, again, in the band cell line. We also saw this in primary brain endothelial cells. Okay, so to conclude, I hope I've been able to convince you that we can harness the low endocytosis of brain endothelial cells to generate artificial targets, but that we need to be careful about which cell membrane component we use to try to establish this strategy. And by shifting our attention from cell membrane components, so cell membrane proteins, which are highly expressed on the brain vasculature as targets, to instead using cell membrane components which have a very low rate of endocytosis, we should be able to expand the repertoire of ligands at our disposal to deliver therapies to the brain. The brain tag, so the strategy we call brain targets artificially generated, or brain tag for short. And it's a new strategy which will hopefully increase the specificity and efficiency of nanoparticle delivery to the brain. And okay, my time has expired, so I will just say thank you to all my collaborators, my funders, and you for listening. Thank you very much.